Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. My name is Safina Abdul-Zubaraba as your Master of Ceremony of today. Alhamdulillahirabbil alamin wa bihi nasta'in ala umuri dunya waddin sayyidina Muhammadin wa ala alihi wa sahbihi ajma'in amma ba'du. The respectful head of Department of Industrial Engineering, Mr. Muhammad Ridwan Andi Purmono, the respectful Secretary of Department of Industrial Engineering, Muhammad Ragil Suryaputro, the respectful Head of Master Program in Industrial Engineering, Mr. Winda Nur Cahyo, the respectful Head of Undergraduate Program in Industrial Engineering, Mr. Dr. Taufik Imawan, the respectful Secretary of Undergraduate Program in Industrial Engineering, Mrs. Sri Indrawati, the respectful Secretary of Undergraduate International Program in Industrial Engineering, Mrs. Ira Prosma Santi Rahmadewi, the respectful Professor Anirban Mukhabadi, the respectful all lecturers in Industrial Engineering International Program, the respectful of all of the participants from Alumni of International Program Industrial Engineering Islamic University of Indonesia, University of Technology Yogyakarta, University of Kalyani, University of Diponegoro, Atmajaya University, University of PGRI, Universitas Bung Hatta, Balikpapan University, University of Indonesia, Gadjah Mada University, Brawijaya University, and many other more. So in this occasion, first thing first, let us pray and praise to Allah Subhanahu wa Ta'ala. Because of His grace, we could attend the second Studium General about the machine learning and biological data mining. And Salat Salam may always to be upon to our Prophet Muhammad Sallallahu Alaihi Wasallam, who brought us from Jahiliyah era to the lightning era. Ladies and gentlemen, to initiate this program, let us recite Basmalah together. Bismillahirrahmanirrahim. And before we open this Studium General, let me read some today's agenda. The first one is opening. The second one is opening speech by the Secretary of Industrial Engineering International Program Department, Mrs. Ira Prosmawanti Rahmadewi. And the second one is about the topic of today, about the machine learning and biological data mining. The next agenda is question and answer, and the last one is closing. So without further ado, please Mrs. Ira Prosmawanti Rahmadewi to give us opening speech for today's Studium's General. Time is yours.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Excellencies, colleagues, ladies and gentlemen, first of all, I would like to say thank you for Allah for the blessing that we are able to join in this webinar. It's really my great pleasure for me to welcome you in the last webinar of IP Industrial Engineering UII Talk on Big Data Series. This is the last webinar hosted by International Program, Department of Industrial Engineering, Universitas Islam Indonesia, in which we are going to discuss about machine learning for biological and social media data mining. Actually, we already conducted several IP, IE, UEE talks, and then they have been successfully conducted. It's really beyond our expectation that a large number of enthusiast participants join our webinar series from all over the world. The first webinar, you already remember, about big data and artificial intelligence issues, in which we know that Indonesian government has established a national strategy of artificial intelligence to keep up with the Industrial Revolution 4.0. First webinar discussed about technology for big data, such as accelerated data science, ecosystem with GPU and rapid, and also the potential research in industrial engineering. And the second webinar also already conducted collaboration between BSN, UII, UGM, UI, Ministry of Health, and underwriter Laboratory USA and also IEEE Indonesia discuss implementation of artificial intelligence in healthcare, telemedicine, telehealth, and standard conformity. And also, we already conduct certified associate in project management, CAPM, and project management implementation webinar. And this is the last webinar in this term. Don't forget, international program, Industrial Engineering, inshallah, will bring out hot issues, the latest issues in business environment, in artificial intelligence era, with other webinar, and we will invite you to join. I hope this webinar will build a strong insight for you to reshape your outstanding better future. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mrs. Ira, for giving us some speech and words for our today's topic. Without further ado, let us introduce you to our moderator today. Her name is Mrs. Anissa Uswatun Hasanah, and she is one of the lecturer in Industrial Engineering Department. She is also the head of Industrial Statistics and Optimization Laboratory. She was born in June 21st, 1990, and her educational background were double degree in fast track master program at National Taiwan University of Science and Technology in Taipei, Taiwan. And she has some technical skills, which is rapid minor, basic coding with C++ language with code block, and also basic MATLAB. So for Mrs. Anissa Uswatun Hasanah, please, I will give you time to host this webinar. Time is yours.
Okay, thank you very much, Mbak Safina, for the introduction. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. The respectful head of Department of Industrial Engineering, Faculty of Industrial Technology, Universitas Islam Indonesia, the respectful head of undergraduate program in Industrial Engineering, International Program, Magister Program, Faculty of Industrial Technology, Universitas Islam Indonesia, the respectful speakers, and as well as all of the participants for this webinar today. Good afternoon, everyone. Alhamdulillah, let us pray and praise to Allah subhanahu wa ta'ala for the bless and mercy. Therefore, we could attend this valuable forum, inshallah. Welcome to our webinar series. And for today, we have special topic for machine learning for biological and social media data mining. It's very glad for me to be here as your moderator, and I'm glad to welcome our special speakers. We have two speakers today. The first speaker is Professor Anirban Mukhopadhyay from University of Kalyani, India. Welcome, Professor. And the second speaker is Mr. Winda Nur Cahyo, PhD. He is from Industrial Engineering, Universitas Islam Indonesia. Before we start the first presentation from Professor Anirban, would you please help us to answer several questions? Your answer will help us to better understanding about your background. Yeah, please kindly find out the questions on your screen. Mas Dwi, tolong. Yeah. For all the participants, please, maybe you just need a minute to answer these questions. Okay, we already have for about 81 participants who have already answered the question. It's about 74% of all the participants. I think it's enough. Thank you very much for your participations. Maswi, would you please help me to show the result? Okay, and this is the result. And so most of you have more than half of you, yeah, from industrial engineering, and then most of you has already know about the machine learning and have ever studied about what is data mining. And yeah, for the answer, 88% from you answer true for these questions. Okay, thank you very much. Your answer give us a general information about your educational background. So for this webinar series, we will learn more about the application of data mining techniques, especially in biological and also in social media that will be presented by our special speakers. And let's begin, or let's initiate our first presentation. But first, let me introduce our first speaker, Professor Anirban Mukhopadhyay, and I will show you his. Okay, this is the biography of Professor Anirban Mukhopadhyay. He is currently a professor of the Department of Computer Science and Engineering, University of Kalani, West Bengal, India. And he's got his bachelor degree from National Institute of Technology, India. And he got his master degree and also PhD in computer science from Jadavpur University. And he is active in several international associations such as IEEE, and then Association for Computing Machinery, and International Association of Engineers in Hong Kong. And his research interest is including soft and evolutionary computing, data mining, multi-objective optimization, pattern recognitions, bioinformatics, and optical networks. And I will give the time for you, professors. Thank you very much.
So hope I'm audible to you. And I would like to, first I would like to thank you for inviting me in this webinar. And today I am going to talk on machine learning applications in biological data mining. So let me first share my presentation. Well, so myself, it's Dr. Anirban Mukhopadhyay. I'm a professor of Department of Computer Science and Engineering of University of Kalani, India. So I am basically a computer scientist, and now we are doing machine learning and data mining for biological data analysis. So today I was checking the voting percentages, and I didn't find anybody to have a biological background. So what I will do that I will first introduce a little bit of biology, and then I will go for what data mining and machine learning can do for analyzing the biological data. So this will be the outline of today's talk. First, I will introduce what bioinformatics is and a little bit of molecular biology concepts. And then I will talk about the different tasks that you can do on biological data sets. And then I will discuss some of the applications of machine learning in biological data mining. And finally, I will conclude. So you see that bioinformatics basically refers to the creation and advancement of algorithms, computational and statistical techniques, and theory to solve formal and practical problems arising from the management and analysis of biological data. So basically what happens that the biological experiments, in wet lab experiments, they generate huge amount of data. The biology is no more restricted in wet labs. These experiments generate huge amount of data, which are needed to be stored in computers. And we need algorithms and statistical and mathematical techniques, programs for analyzing this biological data. So we need applications of statistical techniques, mathematics, and computer science. So essentially, computational biology or bioinformatics is an interdisciplinary field which requires the application of computer science, applied mathematics, and statistics to address the problems inspired by biology. So what I will do, I will just quickly introduce a little bit of biology. Otherwise, it will be difficult to understand what are the applications of machine learning in biology. So more specifically, the molecular biology concepts. So you see that our body is made up of cells, billions of cells, And when we have a collection of cells that usually made a tissue, and when and a collection of tissues made some organs, and finally an organism is basically composed of several organs. And there are two main actors in the chemistry of life. One is the nucleic acids, another are the proteins. And molecular biology research is basically devoted to the understanding the structure and function of these main actors of life, that is nucleic acids and proteins. So let me start with an example. So if we consider a library, we know that within a library we have several bookshelves, and within each bookshelf we have several books. Similarly, if we look into one of our cells, inside our cells we have a nucleus, and within the nucleus we have several chromosomes, which can be considered as the bookshelves of the library. So if the nucleus is the library, then chromosomes are the bookshelves. And within the bookshelves, as there are several books, similarly within a single chromosome, it consists of several genes. And the astonishing thing is that almost every cell in an organism contains the same library and the same set of books. That means all the cells have the same set of chromosomes, and so we have the same set of genes in every cell of our body. Now, these
I'm sorry, Professor.
Yes.
I think your slide has not changed.
Oh, so is it changing now?
Not yet. It's still on the first page.
Oh, sorry.
Oh, yes. Yes, now it's already changed. Thank you very much.
Let me keep it like this.
Yeah.
So you see that these chromosomes are basically made of some DNA molecules, deoxyribonucleic acid. I would like to discuss a little bit of the structure of the DNA, because that will be helpful. So before that, let us discuss some of the terminologies that will be helpful for you for understanding what molecular biology is. So genome is an organism genetic material. That means if we consider all genetic materials of an organism, that's what we call as genome. Gene is a discrete unit of hereditary information. This is located on the chromosomes, and they consist of DNA. So what we have a chromosomes. Chromosomes are thread-like structures, and within a single chromosome there are several genes, and each gene, so the whole chromosome is actually made of DNA. So obviously each gene is also made of DNA. Genotype means the genetic makeup of an organism, what is inside in your cells, what are the structures of the chromosome, what are the genes you have. So that is your genotype. And phenotype is physical appearance. That means what we see you from the outside, how you look like, how your behavior is, what is your intellect. So all these is your phenotype. And nucleic acids, that is, there are mainly two types of nucleic acids, RNA and DNA. So DNA is used, DNA is actually makes your chromosomes and the genes. And what is the use of RNA, or what is the responsibility of RNA, I will discuss a little bit later. So this DNA and RNA actually allow the organism to reproduce. The genome is an organism's complete set of DNA, as I mentioned, the complete genetic material. And human genomes have approximately 3 billion base pairs. What is base pair? Actually, you can consider as a string. DNA is a string, and there are 3 billion characters in a human genome. And human genome also has 23 pairs of chromosomes. So inside each cell of our body, there are 23 pairs of chromosomes, except our sex cells, where we have exactly 23 chromosomes, 22 autosomes and 1 sex chromosomes. So for males, we have X and Y, and for females, we have 2 X chromosomes inside our cells. So gene is basic physical and functional units of heredity. So genes are specific sequences of DNA bases that encode the instructions on how to make proteins. So basically, genes contain the program how a cell will function, how an organism will function. The whole program actually resides in the genes. But the genes themselves cannot function. In order to execute that program, genes are to be translated into a different molecule which are called as proteins. So proteins are products of genes. They are basically large and complex molecules made up of smaller subunits called amino acids. So genes are made of the nucleic acids, basically because they are basically made of DNA, and proteins are made of amino acids. So from computer science perspective, if you have studied operating system, there are two terms, you know, that one is called the program and another is the process. So what is a program? Program is basically when we write using some programming language, we write a program and that resides in the disk. So program is the code, but they cannot execute until you that program cannot function until you execute the program. So once the program is executed, the program comes in the memory, in the RAM, and then we call them as process. This process actually executes the executable version of the program. It is the program in execution, we can say. So similarly here, the genes are the programs, but they themselves cannot function, although they have all the information how the cells should function, but they themselves cannot function. So we have to convert the genes into proteins, and these proteins are the functional units which can execute the program which are residing in the genes. So it is very important. So the whole molecular biology concepts actually stand on the fact that how the genes are converted into the proteins. So that is called the central dogma of molecular biology. I will a few slides later, I will discuss that in a little bit more detail. So we have to deal with 3 molecules, DNA. So out of them, 2 are nucleic acids, DNA and RNAs. So DNAs made genes, so they hold information on how cell works. And RNAs actually is an intermediate thing between the DNA and proteins. So while DNAs or genes make proteins, they have to pass through a state which is called an RNA. So RNAs act to transfer short pieces of information to different parts of cells. So sometimes it is called the messenger RNA, that's why. And they provide the templates to synthesize into proteins. As I said, that they are intermediate molecule between the DNA and proteins. And proteins are basically, they have many jobs inside the cells. They make enzymes, they make hormones, they carry signals from one part of the cell to another or from one cell to another. And they also form body's major components like hair, skin, etc. So all are made of proteins. So that's why making proteins is very, very important. Although the genes have the program, but until you make them process, that is the protein, your cells cannot work. So this is the overall visualization of what I have already talked to you. That is, inside our cells, we have a nucleus. Inside the nucleus, we have several chromosomes. If we consider a single chromosome, that chromosome is basically made of DNA molecules. So this is a DNA structure. This is called a double helix structure. I will just show you in what actually it contains, this double helix structure. And as I said, that in a single DNA molecule or in a single chromosome, there are several genes. Some parts, some sections of the chromosomes are the genes. So if we look into the DNA molecule, actually, this is very important. This is a double helix structure. Inside that, the DNA molecule actually consists of two chains, two strings, and each string, each sequence is made of basically 4 nucleotide bases, which is called A, T, C, and G. They stands for adenine, thymine, cytosine, and guanine. And each each string of the DNA is made of a arbitrary sequence of A, T, C, and G. And if you know one strand or one string of the DNA, you can automatically find what is the sequence of the other string, because A always pairs with T. So if one strand there is A, in the opposite strand there will be definitely a T. And C pairs with G. So if one strand there is C, then in the opposite strand there will be G. So it is sufficient to know one strand, the sequence of one strand of the DNA, because the other strand can be automatically found out. And this entire genetic information is stored in the DNA strand. So if we look, so as I said that these are the nucleotide bases, 4 nucleotide bases. If we look into the nucleotide bases, I will just quickly go through this. I will not go into very details of biology, but little bit. So each nucleotide base actually looks like this structure. There will be a pentose ring, a phosphate. And where each nucleotide bases are different is that in this part. So that base is connected here. So A, T, G, and C will have difference here. And what is the difference between DNA and RNA? If we look into this pentose ring, there are basically these 5 carbons are there. 1, 2, 3, 4, and 5. They are numbered clockwise. 1, 2, 3, 4, and 5. And in DNA, the second carbon has an hydroxyl group, and in the RNA, the second carbon has the hydrogen. So that's why it is called. So when we remove the oxygen, so from the hydroxyl we get the hydrogen. So that's why it is called deoxyribose nucleic acid. So this is the DNA where we have hydrogen. So we don't have oxygen. So we have deoxyribose. So that's why it is DNA and it is RNA. So as I already mentioned that in DNA, A pairs with T and C pairs with G. And in RNA, there is a little bit of difference with the DNA is that instead of T, RNA has U. So instead of thymine, RNA has uracil. Otherwise, the other 3 bases are same. So in RNA, A will pair with U and C will pair with G. So this is what the DNA molecule looks like. 2 strands, as I already mentioned. This is one strand and this is another strand. And each strand is basically a sequence of A, T, C, and G. So this continues. And if you know the sequence of one strand, you automatically know the sequence of the other strand, because A always pairs with T and C always pairs with G. So this is what I was telling you, the central dogma of molecular biology. This is the process of making proteins from genes or DNAs. So this is the two-step process. The first step is known as the transcription. By transcription, a DNA molecule is converted into an RNA molecule, especially it is called a messenger RNA because this carries the message of the template of the proteins. And the second step is called translation, by which an mRNA is converted into a protein. So DNAs are the sequence of nucleic acids. As I said, these are basically sequence of A, T, G, and C, any random sequence. And when DNA is transcribed into mRNA, we call this phenomena as gene expression. We call that a particular gene is expressed. And when mRNA is converted into protein, that is known as translation. And this translation actually creates protein, which are nothing but a sequence of amino acids. There are 20 different amino acids in nature. And how this translation actually happens? This translation actually happens inside the ribosome of your cells. And if you look into this RNA structure, see, this RNA is nothing but one particular strand of the DNA, and all the T's are replaced by U's. As I said, that RNA does not have thymine, instead they have uracil. So the T's are replaced by U's, and each triplet of nucleotide bases, for example, CCU, then GAG, then CCA. So each triplet is translated into one amino acid. So CCU translated to P, GAG translated to E, like this. So we use 20 letters of English alphabet to represent the 20 amino acids. So this way, each triplet, which is also known as codon, so each codon is translated into one amino acid. So this is a sequence of amino acids which actually creates the proteins. Now, I will not go into much details at what is actually happens, how this actually happens. So that is more biological, but you have to at least understand that this process of central dogma of molecular biology, that DNA converts into mRNA and mRNA converts into protein. I would like to discuss two things here. So DNA means, you know, the genes. So this is the process how the gene is converted into protein. Now, I mentioned before, if you remember, that all the cells in our body have the same set of genes, almost same set of genes. So as the genes are the program, we can say that all the cells in our body have the same program. Then the question is, why the different types of cells, for example, why the function of heart cells are defined are different from the functions of a liver cell, and which is again different from the functions of a lung cell. So why the cells of different tissue types have different functions, although they have the same program? Because although they have the same set of genes, not all the genes are expressed in all types of cells. Expressed means that not all the genes are creating mRNAs, not all the genes are transcribed into RNAs. So this phenomena is known as differential gene expression, where by which the different types of cells have different subsets of genes which are expressed in different types of cells. And that's why different subsets of proteins are created in different types of cells. So the proteins which are created in heart cell are not necessarily same as the proteins which are created as in the lung cells. So that's why the functions of different tissue types and different types of cells are different. This is very important. This is called the differential gene expression phenomenon. These are the details of transcription and translation. I'll just skip this part quickly. So what I said, that each codon, that is each triplet of nucleotides in the RNA sequence, is converted into one amino acids. It's translated into one amino acids. And the mapping between these codons and the amino acids, this is very important. This is the central thing of our life. Because of this code, actually, we are alive, we can say, and we are functioning. So this is called the genetic code. So genetic code is basically a table that maps each codon or each triplet of amino acid, each triplet of nucleotides into one amino acids. So this table actually gives you the genetic code. So obviously you see that how many codons are possible, as there are 4 nucleotide bases and we have 3 positions. Triplet means 3 positions. So first position can consist of 4, the second position can also consist of 4, and third position also consists of 4 possible nucleotides. So we have total 64 possible codons. And as I mentioned, we have 20 amino acids. So this table actually gives you the mapping between 64 codons and 20 amino acids. So obviously you see that this is not an 1:1 mapping. So multiple codons can encode the same amino acids. So that's why, although the RNA sequence may be same, but sometimes the different proteins are created. So once the proteins are formed, they are just a polymer of amino acids, a sequence of amino acids, a long chain of amino acids. And proteins are the functional units, as I mentioned. So they have several functions in the cells, like they make enzymes that rearrange the chemical bonds. They carry signals to and from the outside of the cell and within the cell. They transport small molecules, like hemoglobin is a protein, and these proteins transport the oxygen molecules, inside our bloods. The proteins form many of the cellular structures, the cell membranes, the cellular structures membranes, right? The ribosome, the Golgi bodies, or mitochondria. So different parts of the cells are also made of the proteins. And they regulate cell process. That means, as I mentioned, the differential gene expression, not all the genes are expressed in all the cells. So which genes will be expressed, that are also controlled by a special kind of proteins which are known as transcription factors. Because these proteins decide or regulate which genes will be transcribed and how much transcription will be done for those genes. So that's why they are called the transcription factors, and they are nothing but some proteins. So when the proteins are first formed, they are simply a sequence, a one-dimensional sequence of amino acids. But actually, in order to function, the proteins start folding to give a 3D shape, and they have gradual foldings. So the first formed protein, that is the sequence of amino acid, is called the primary structure. Then after a little bit of folding, they create a secondary structure, 3D structure, and then tertiary structure, and finally multiple protein peptide chains are formed together to form the quaternary structure. This is the most complex structural form of 3D structural form of the proteins. And the proteins, ultimately what functions will be performed by the protein depends on its final 3D structure. So so after this, so this is what the molecular biology concepts you actually have to understand in order to work for biological data. So you see that what is our data here? So our data are mainly in earlier days, our data was only the sequences. So you know that all the biological molecules are basically made of sequences, like the DNA and RNAs are nucleotide sequences, the proteins are amino acid sequences. And there are large number of DNA, RNA, and proteins inside our cells. So the amount of data that is these sequences is huge. So that is one kind of data you have. But modern biology can give rise of other kind of data like expression data. I mean that this is the data of which genes are expressed and how much they are expressed, that value, expression value of the genes in different types of cells or even each cell in the body. That is called the expression data. And also we have to deal with the biological networks, because the biological molecules, they rarely function as individual units. Instead, the different biological molecules actually interacts with themselves, like the DNAs interact with proteins, proteins interact within themselves, and there are other metabolites, they interact with each other. Thus, they give rise to different networks inside the cells, where the nodes of the network are the biological molecules, and the edges represent the interactions between them. So the biological networks are also a kind of data which are hugely popular in the machine learning domains and data mining domain. So we have to deal with graph data then, because the networks are represented as graphs in computers. So here are some examples of different common proteins, like protein functions, like they act as hormones. For example, insulin, you know that is a hormone used for treating the blood sugars. So this is a protein, insulin is a protein. The protein channels, so protein channels actually used for transferring some substrate from one part of the cell or from inside the cells to outside and vice versa. And as I mentioned, the transport of molecules. So hemoglobin is a protein in red blood cells, and they transport oxygen molecules from one cell to another. So let me just now discuss that what are the different computational tasks or data mining tasks that you can expect in bioinformatics or computational biology. So we can categorize this task in mainly 4 different levels. One is the sequence level task that basically deals with the sequence data, nucleotide sequences or amino acid sequences. Then we have the structure level tasks, which deal with the 3D structures. As I mentioned, that proteins are folded into 3D structure in order to function. And when we have to deal with the structures of the proteins, 3D structure of the protein, that is our data, then we have several structural level tasks. Then expression level tasks that deals with the gene expression values. So our data here are the gene expression values. And finally, the system level tasks, which deal with the biological networks, because biological network gives you the idea, understanding of the complete system. The whole system or whole cell is functioning. What is the interaction between the different molecules within a cell? So that's why they are called the system level tasks. So under the sequence level tasks, we have several subtasks like the sequencing the genome. Sequencing the genome means what is the exact sequence of ATGC in your genome. That is very important, because nowadays you have probably heard one particular term that is called personalized medicine. They have seen that it may happen that 2 persons have the same disease, but the same medicine do not work for them. The same medicine does not work for them because their genetic makeup is different, their genotype is different. So in order to find out which particular medicine will be effective for a person, it is very important to know that what exact sequence of genome or what is the exact sequence of ATGC is in your genome. So it might happen that in near future that the doctors will ask you just when you go to the doctor, and nowadays the doctors ask you to do some tests like some blood tests or some urine test and so on. And one day will come that when you go to a doctor and the doctor will ask you that, okay, you go to a diagnostic center and sequence your genome. And you come with that sequence, and according to that sequence, the doctor will prescribe medicines. So actually, the sequencing the genome is more like a computational task, because the genome sequence is very large. No sequencing machine is available to that sequence that genome in a single run. So what is usually done, the whole genome is cut into pieces and each piece is sequenced. So these are called the fragments. And finally, these fragments are computationally assembled with the help of some reference genome, and to find the whole sequence of your genome. Then sequence alignment, that usually used for comparison of sequences from different species. This is very important to know, generate that phylogenetic trees or evolutionary trees to know that relationship or evolutionary relationships of different species. Like, for example, you want to know the relationship between the humans and monkeys and baboons and gorillas and so on. And you can compare their sequences to build a evolutionary tree structure to know that at which point of time in history we have actually deviated from each other. Each species is deviated from the others. That can be done. So basically, we have to apply some kind of clustering techniques for more specifically the hierarchical clustering techniques for that purpose. Gene finding and promoter identification. So in which parts of the sequence are the genes? As I mentioned, that not the whole DNA sequence is a gene. Some sections of the DNA sequence is a gene. And it is important to know that which sections are genes. So the gene finding, because the genes may have some special sequences in the genes, so special pattern in the sequences. So we have to identify those patterns. So this is basically a pattern recognition task from the sequences. And protein superfamily classification. So whether we can classify the protein based on their sequences, amino acid sequences. So these are all sequence level tasks. Then we move to the structure level tasks. So under this, we have to protein structure prediction. Like you were just given a amino acid sequence of proteins. So whether you can predict the 3D structure from this single dimension sequence of the proteins. So for that, we have to basically predict that how the protein actually folds. So all these are computational tasks. They are mainly posed as optimization problems, and different optimization algorithms can be utilized for that. Structural alignment, we have done sequence alignment. Similarly, we can do structural alignment to know that, to compare the structures of two or multiple proteins. So how they are structurally similar. Then structure-based protein classification. So we can classify the proteins based on the structure. So from the protein structures, we have to basically extract some features, and we have to train a classifier based on those features. And finally, we have to classify the proteins based on their structures. And molecular docking, molecular design and docking. So that is another task in the 3D structures, in order to deal with the 3D structures, that whether how the two proteins will interact and in which part the two proteins will interact, and what may be the possible interaction site between a drug and a protein, a medicine and a protein. So all these are the part of molecular design and docking. Then we have to study the expression changes of the genes. As I mentioned, that expression levels of the genes, different cells have different genes have different expression levels in different cells. So we have a technology called microarray technology, which actually helps you to monitor the expression level of thousands of genes in a single data sets. under different experimental conditions. So these expression data are basically represented as matrices, and we can do several data mining tasks on this expression data, like clustering of genes in order to know that which genes are similar based on their expression pattern over the different experimental conditions. Then classification of expression data, whether we can classify the different cell types, for example, normal cell types and malignant cell types for a cancer disease, or for other kind of diseases, whether we can differentiate the cell types based on the expression pattern of the genes in the cells. Gene marker identification, that means which genes are uh, which genes have the ability to distinguish the different cell types. So these are known as the gene markers. So these are mainly posed as the feature selection tasks, and we can definitely use some data mining and machine learning techniques for doing this kind of feature selection. Then we have to deal with the system level tasks. We have different biological networks like gene regulatory network, which are basically interaction between the genes and the transcription factors. Then the metabolic networks, the interaction between the different metabolites inside the cells. Protein-protein interaction network, the interactions between the different proteins. The proteins normally do not function individually, rather they function as a group, so they interact with each other. So all these networks are basically represented in computers as graphs. So the vertices of the graphs are represented as, they represent the different molecules. For example, in protein-protein interaction network, these vertices represent the proteins, and the edges represent the possible interactions. And we can apply the different graph theory algorithms and graph data mining tasks on this network data in order to, for several tasks like identifying network biomarkers or prediction of new interactions, identifying the dense region of the networks like protein complexes, and so on. So, and what are the main actors inside the network? That means what are the main or the key nodes or key vertices in the network. So all these are part of this graph data mining tasks. And there are some other kind of tasks like the study of drug response. That means that whether a drug will respond, whether a patient will respond to a drug or not. Then drug schedule optimization, you can do. These are not directly related to bioinformatics or other, but drug schedule optimization can depend on your genetic makeup. So that is one area of personalized medicines. Then survival prediction, whether you can predict the survival time of a particular patient, and what are the different genetic factors behind that survival analysis. Cancer prediction, whether you can predict the cancer genes or cancer tissues, so they can help in diagnosis. So these are the different other tasks under computational biology and data mining of biological data. So I will finish now quickly. So what you see that impacts of bioinformatics. So bioinformatics classically was a wet lab science. That means the people did experiments in the wet labs, but with the biology was basically wet lab science, but with the invent of bioinformatics, it now leads to the advances in understanding the basic biological process, treatment, and prevention of many genetic diseases. That the bioinformatics helps a lot in these respects, starting from any kind of diseases, like it can be genetic diseases like cancer, or it can be communicable diseases like viral infections. Recently, that due to the, that the COVID pandemic, a lot of bioinformatics works have been done in order to understand that virus SARS-CoV-2 with the help of bioinformatic tools. and definitely machine learning and data mining on the data, biological data. So bioinformatics has transformed the discipline of biology from a purely lab-based science to an information science as well. So it is no more a wet lab-based science only. So it is also an information science. And modern biology and related science are increasingly becoming dependent on the bioinformatics or computer science. So bioinformatics exhibits great potential in future development of science and technology, definitely. So I will just touch a little bit of machine learning and what are the different machine learning works on biological data that we are doing in our labs. So you know that those who have studied machine learning or data mining, you know that there are machine learning is basically a field of computer science that helps systems to learn automatically from the given data without being explicitly programmed. And there are mainly two types of machine learning tasks, descriptive and predictive, also known as unsupervised and supervised. So under unsupervised tasks, we have clustering, association, rule learning, and so on. And under the predictive tasks, we have classification or regression analysis. So the basic difference is that in the unsupervised tasks, we are just given the raw data and we don't have any training set through which we can train our algorithms. We have to just use that data and we have to extract knowledge out of that data. And predictive or supervised tasks, they generally have some training sets. With that training data, we have to train our classifiers or regressors. And finally, we can apply those trained classifiers or regressors to do some kind of predictions from out of new data. Dimensionality reductions or feature selection, they can be both supervised or unsupervised. So these are the different machine learning tasks. And in our labs, we are doing many machine learning works on with the applications in bioinformatics and biological data mining. Like we are doing clustering and classification of gene expression data, as I mentioned. Then we are also working on gene marker identification from the gene expression data. Which genes are important, distinguishing markers in order to distinguish the different disease subtypes. So these are called the gene markers. Then we are also doing graph theory works on analyzing biological networks. So basically we represent the networks as graphs and then we apply different graph algorithms and graph data mining tools on these biological networks in order to extract several important knowledge like which biological molecules work together, like we identify the protein complexes. Then we also perform protein-protein interaction prediction, mostly in host pathogen interaction or viral host interaction, that which viral proteins interact with which human proteins. This is very important because unless we can identify the possible interactions, we cannot find our targets, drug targets. Basically, these drugs actually target these interactions to break these possible interactions between the viral proteins and the human proteins. So unless we don't know which proteins, which viral protein interact with which human proteins, it is not possible to find out the drug targets and design the corresponding drugs. And multi-omics data analysis, this is recently we are doing because the biological data have for the same patient, suppose we can collect the data from different omics in the sense that we have genomics data, we have proteomics data, we have metabolomics data, we have other kind of demographic data. So we have different types of data for the same set of patients, and we are analyzing how to integrate this data from different aspects, from different views, and finally get something out of it that is known as the multi-omics data analysis. So these are the different machine learning and data mining tasks we are doing on the biological data. Hope you have got some idea of what machine learning can do for the biology. Uh, so I will now conclude because I think the time is almost over. So in conclusion, I can say that there are several challenges in biological data mining. You see that sometimes we have huge amount of data. For example, genomic sequences data, especially with the advent of next-gen sequencing techniques. So we have a lot of sequence data available. So it may be in order of gigabytes or even terabytes. So we need very fast algorithms, data mining algorithms for analyzing this sequence data. Then expression data of genes with the advent of single cell expression. So previously we could not uh, have enough resolution to measure the expression of genes from each cell, but with the advent of new technologies, single cell technologies, we can measure the expression level of each gene in each cell. So that's why we have the matrix of expression data. It's huge because we have thousands of genes, like maybe we have 30,000 genes in one side, and we have some lakhs of some millions of cells in the other dimension. So it's a huge data. So for analyzing huge data, we need very efficient algorithm, time efficient as well as space efficient algorithms, data mining algorithms. That is one challenge. Sometimes another challenge is lack of data. In some aspects, in some kind of data, for example, small RNA related data. So RNA related data is especially that non-coding RNAs. So messenger RNAs are coding RNAs because they are carrying the gene signals to convert into proteins, but there are other types of RNA as well, like microRNA is there, the small RNAs are there. So the data related to these RNAs are very small. So sometimes due to the lack of data, we sometimes have a problem in analyzing the data, or we have the problem in developing our training sets because of in the machine learning task because of lack of data. Then we have noisy data. The biological experiments they generate data sets with noises. So when we apply our machine learning tasks on these data sets, we have to denoise these data sets. So this is this problem. And meshing data. So also that these data sets have different several meshing values. So we have to deal with this meshing data. We can have to deal with some missing value estimation techniques and so on. So these are the different challenges that we have to deal with when we have to perform these biological data mining tasks. So that's all from my side for now. So this is the book we had written. This is um, actually the multi-objective genetic algorithm for clustering. We have shown that application of this clustering on the bioinformatics tasks. And these are some of the references from my own papers. And if you are interested, you can just look into my site. Or if you have any question later on, you can also mail me. So I'll be happy to answer. So thank you for your patience, and hope you have enjoyed. Thank you.
Okay. Thank you very much, Professor, for your very interesting presentation. Professor Anirban has already explained to us about the application of machine learning in bioinformatics. And I think Professor Anirban has also explained there are a lot of function of machine learning in bioinformatics, such as to classify cell cancer that can help doctor to predict the cancer disease, and also to cluster the gene data, or to predict protein interaction and so on. And I think this kind of application of data mining can further can help medical world to help doctor to diagnose some disease or give a suitable treatment, or also can prevent many genetic disease. Okay, one more time, thank you very much, Professor, for your presentation. And now is it question and answer session. And we already have several questions for the participants. I will read the questions and Professor Anirban will help me to answer the questions. For the first question, it is from Masitoh Yesi Rohayani. And the question is, I am interested in analyzing RNA sequence data. I have read some papers, but I don't understand the term of read count. What is read count? Maybe Professor Anirban can answer it first.
I'm not sure because I don't work in the RNA sequence data. So at this moment particularly, I cannot answer. So you please send me a mail. I will check and we'll get back to you.
Okay. Okay. Mrs. Masitoh, would you please send Professor Anirban an email, yeah, for complete explanation from him. And for the second question, Professor, this is from Mr. Maji. How can we find the gene sequence from the DNA using these machine learning techniques?
That's a good question. Actually, you see that as I mentioned that the chromosome is made of DNA, the whole DNA sequence, and only some sections of the DNA are the genes, right? So generally what happens, these sections, these particular, the gene sections of the DNA, they may have some particular pattern in the sequence. And we have to basically identify these patterns, that which part of the DNA has these patterns. And that is usually a pattern recognition tasks. And we have the long string of DNA sequence. So it's a huge data. And out of that, we have to identify those sections that has that particular pattern. So we have different algorithms for that. I think we don't have enough time to discuss all these things, but this is essentially pattern recognition tasks, and we have to identify particular pattern, particular motifs out of these whole DNA sequences, and those particular motifs or those particular sections will be our genes.
Okay, thank you, Professor. And for the third question, this is from Muhammad Iqbal. And the question is, should data scientists be good at mathematics to implement machine learning algorithms, especially in biological data mining, and how to get digital data?
The first part of question, that whether a data scientist must know mathematics, I will say yes. In order to become a very good data scientist, you have to understand the mathematics as well as the statistical techniques, because nowadays there are several programming tools are available, and people are doing data scientists work without even knowing what is what happening inside. But you will be a better data scientist definitely if you know what is happening inside, how the data science tools are actually working, what is the mathematics behind that. Then you will be able to apply these tools more wisely. And second part, where you get this data, actually from where you can get this data. Actually there are for accessing the biological data, there are several public repositories available. If you are interested in any particular kind of data, biological data, just drop me a mail. I will send you some links from where you can access this public data. Okay, and but obviously, if you want to do some more real data analysis tasks, you have to collaborate with some biologists. They will generate data from their labs, and you will have to analyze that data. But there are definitely a lot of public repositories available which have repositories for real data sets.
Okay, thank you very much, Professor. And thank you very much for all the participants that have already given questions. And the next question is from me, Professor, because I also work in data mining and I do several research in data mining. And as we know that when we're dealing with a lot of data, one process that is it. Sometimes it's difficult to be done is the pre-processing data, and you have already mentioned that sometimes you're dealing with the noise data or missing data and so on. And if for you, how you dealing with this kind of problems, how you deal with the missing data or noise data?
Actually, it depends on the what kind of data we are analyzing. For example, for missing data, if we just consider a gene expression matrix, there may be several missing values out of these expression data sets. So first we look into that how many missing values are there, maybe what percentage of genes have the missing values, or what percentage of cells have these missing expression values. So in either way, if we find that this percentage is very low, and for our task, summarization task, not all the genes are important or not all the cells are important, then if percent is very low, we can simply opt out those genes and or those rows and columns out of this data. That is one aspect. That, but when you have this percentage is large, that means the missing data percentage is large. It's suppose it's almost coming as 50% of data is missing. So definitely in that case, you cannot just remove those rows and columns because you will probably lose a lot of information if you just remove those columns. So in that case, definitely you have to take some measure in order to estimate the missing values. That what may be the possible missing values, what may be the values you can put under those missing columns or rows. And there are different missing value estimation techniques. You can just look into one simple way is just averaging the other column values onto that particular column. That is one possibility, but that is not a very wise thing. So sometimes what we do, a more better technique will be to this nearest neighbor techniques. That means if we just want to find the missing value from a particular cell, we find out that based on the values which are already present, we find what are the other neighboring genes, other similar genes. And we find that out of those similar genes, what is the value of the expression in that particular cell for those genes. And we then average those values for this particular gene for which we have the missing value. So these are just simple some techniques, but there are definitely other more sophisticated techniques to analyze this or to estimate these missing values. And you can just explore that. There are different techniques.
And in your experience, what is for the beginner? Yeah, for beginner, what kind of method that is easy and suitable for us to define the number of cluster if you use a clustering analysis? Because sometimes if we use hierarchy or non-hierarchy, it's sometimes difficult to define how many is the suitable number of cluster.
It's a this is a question of long back. I mean that it's what should be the number of clusters. So normally what we do, we take the help of different cluster validity index, and cluster validity indexes are there are some mathematical expression that actually tries to find out the goodness of a clustering solution. That how good is a clustering solution? Can I come out with some value, some metric value using this cluster validity index? So there are several cluster validity indices, and different cluster validity indices are there. For example, I give you one example called Xi-Veni index. So this cluster validity index take care of mainly two things. What is the cluster compactness? That means what is the within cluster variance, and what is the inter-cluster distances. So basically, we try to minimize the intra-cluster distances within the cluster in order to increase the compactness, and also we want to maximize the inter-cluster distances. So in order to have the well-separated clusters. Now, different cluster validity indexes actually computes these two things in different ways, but basically they try to combine these two values to come out with a single value. Now, what we can do, is that suppose we take a particular cluster validity index, and then we, if you are doing an hierarchical clustering, you have already built a dendrogram, suppose, and the hierarchical tree structure, and you can cut this dendrogram at different levels to have different number of clusters. And for each such different number of clusters, you compute the value of that particular validity index you are using. And suppose you can just try with, normally what we do, if there are n number of data points, we can try from number of cluster from 2 to root over of n. This is just a hard and fast rule, but this is, I will not say that. But if you have some idea that this may be a maximum number of clusters, then you can use that. So suppose from 2 to 10, we just find what are the what is the clustering for, if the cluster number of clusters is 2, what is the clustering, what if the number of clusters is 3, and so on. And then you compute the cluster validity index value for each such solution for different number of clusters. And then choose that particular number of cluster for which you get the best value of the cluster validity index. Best value in the sense that minimum value for the intra-cluster distances and maximum value for the inter-cluster distances. So generally they are combined. Suppose if they are combined as in the numerator, we keep the intra-cluster distance, and the denominator we keep the inter-cluster distances. So the overall, the cluster validity index should be minimized in order to get the compact and well-separated clusters. So out of this, from 2 to 10 different number of clusters, we do the clustering, and then we compute the cluster validity index value for this each number of clusters. And then we choose that number of clusters for which the validity index value is minimum for our case. So that will be the best number of clusters you can expect. This is one technique that generally we follow.
Okay. Okay, thank you very much, Professor, for your answer, and thank you very much for answering all of the questions. And it's great to have your presentation. It's a pleasure to have you with us today. And this is the end of the first session of our webinar. And thank you for all the attendees for your attention. Before we move to the second webinar, the webinar will be continued with the quiz, and the quiz will be led by our Master of Ceremony. Now we go back to Mbak Safina. The time is yours.
Okay, thank you, Mrs. Anissa, for the time. Please, maybe the committee could share their screen so our participants today could join the quiz for today. So please go to the website as you can see on the screen. You can use your mobile phone, PC, or laptop. Just type in joinmyquiz.com with the entering code as shown in the screen 45497 and so. We'll wait for maybe 80 participants to join. And if you have any kind of difficulties when you are trying to join, please let us know. And please use the name that is related to your name on your email, so it will be easy for us to recognize your data. And the top 3 who can answer the quiz correctly and quickly will be having an e-wallet that we will be provided after this webinar is end for top 3. Okay, let's wait for the others. There are already 25. As I mentioned again, if you're having any kind of difficulties to join this quiz, let us know. Okay, there are already around 68 participants. Maybe we could start this quiz. Okay, please start. Okay, I hope you answered the questions correctly and quickly. The question is based on the materials that already provided by the Professor Adiban Mukabadi. And as you can see on the screen on the right side of me, it's the number if you answer the questions not correct or false. And on my left side is the green one is you could answer the questions correctly. So let's see who is the top 3 of this. And for the top 3, please make sure that you give you could contact me or just chat to all panelists your name and also your phone number so we could contact us. And please make sure that you are typing the correct phone number, because if you're missing some numbers, it will be wrong and we cannot contact you. Okay, let's see over here. As you can see on this screen already, there are some points on the runner-up. Okay, let's wait for everyone who are already fulfilled all of the questions. Okay, maybe another more minutes until you guys all fill in the questions on this quiz, and we could have our winner for today's. All right. Maybe we're waiting for another because there's some still blank space over that, the black one, to make it already fulfilled. Okay, I think it's done. So for top 3, please make sure that please give us your phone number on this chat box Zoom meeting, so it will be easy for us. Thank you. Maybe I will hand back to Mrs. Anissa Uswatun Hasanah to deliver the introduction to the second speakers. Time is yours.
Okay, thank you very much, Mbak Safina. And congratulations for all the winners. And now let's begin our second session. But first of all, I would like to introduce our second speaker first. Okay, for our second speakers, Mr. Winda Nur Cahyo, PhD, is our head department of master program in industrial engineering, Fakultas Teknologi Industri, Universitas Islam Indonesia. He got his PhD from University of Wollongong, Australia, and his research interest is including asset management system analysis and design, productivity analysis in industrial management, and management information system decision support and expert system. And for today's agenda, he will the time is yours.
Okay, thank you, Anissa. Can you hear my voice clearly, or?
Yes, it's clear,
Okay,
Pak Winda.
Okay, let me share my screen first for a while. Just give me 1 minute. Okay, thank you for a very good opportunity, and thank you also for having me here in this webinar. And first of all, I would like to apologize to Bulra and all the committee that in the last minute I changed my topic. So the topic that I sent to the committee last time, as I remember, is only, what is it, social media data mining. But in the last minute, I'm thinking of something else, but still related with the social media data mining actually. I want to see the social media data mining from another perspective. Yeah, not about the technical aspect of the social media data mining, but I'm seeing that there is another aspect that it could be more beneficial, especially to the student. Based on that, consideration, in last minute, I changed the topics into encouraging excellence through data literacy. So with this topic, what I would like to conclude by the end of the presentation is how students or anyone who attend this webinar have a bigger awareness of how we can use data. All right, so let's start from the very simple thing. Just give me 1 minute. I would like to introduce myself. That has been done by Mrs. Anissa, Bu Anissa. So I can skip it actually. So, but something that I want to highlight is actually my bachelor degree, as you know, from Industrial Engineering, UI from University Islam Indonesia, and master degree from electrical engineering, in computation and informatics system, University of Mada. And my PhD, as Bu Anissa already mentioned, from University of Wollongong, Australia. All right. Okay. Let's talk first about what is literacy. As you remember that in the subject, I mentioned about encouraging excellence through data literacy. So what literacy actually? So when you try to find the operational definition of literacy, yeah, I try to find it in the Merriam-Webster dictionary online. Okay, so the literacy actually, according to the dictionary, is the quality or state of being literate. The quality or the state of being literate. So what's literate? And then I find in the other page of the Merriam-Webster dictionary, and literate is actually educated, or sorry, there are lots of chat here that closing the screen actually. So literate is actually having knowledge or competence. So in that sense, if I can develop my own definition of data literacy, so data literacy is, okay, data literacy is actually, having a quality or the quality or state of being, having high knowledge or competence in the data. Yeah, so that's my own definition of data literacy. So if you check on the World Economic Forum that has been published not so long ago, maybe in few weeks or few months ago, there are actually four literacies for a better world. There are 4 literacy for a better world. The first one is future literacy. So future literacy is actually the skill that allow people to have a better imagination and make sense of the future. All right. And the second one is system thinking. I believe that any of you who studied industrial engineering or studying industrial engineering system thinking is very familiar. So system thinking is actually a mindset to think, communicate, and learn about system to make a full patterns clearer, improve and share the understanding of problem and see how to face them effectively. So actually system thinking is a mindset to think and communicate about what happened in the system and see the full pattern about what actually happened in the system. And the third literacy for the better world, the third one is anticipation, and the fourth one is strategic foresight. I will not discuss those two in detail, but you can read there, and I will send these slides to the committee, yeah, and you can request from me. So if we talk about literacy, and currently we are in the system of higher education, so in a system, actually, as you know, there is an input, there is a process, and there is an output. Okay, and higher education system is actually a system to process the input, which is maybe a student from high school, yeah, into a predetermined output. In my sense, the output, one of the skill that we need to provide to the graduate is how they can be literate or being literate. Okay, but the issue is what kind of literacy that they need, especially in facing the environment of the surrounding system, because the output of the higher education means that they have to go back to the community, they have to go back to the community, to the society. And the society now facing a very big challenge, especially as you know, like issue related with industrial revolution 4.0, issue related with society 5.0, and something like that. Okay, if we see further, what's actually industrial revolution 4.0, and I believe that most of you are already familiar with industrial revolution 4.0. There are 9 pillars of industrial revolution 4.0. And among those 9 pillars, what I'm focusing on this discussion is actually the big data analytics, the big data itself. Big data is one of the pillars of industrial revolution 4.0. Okay, so the next question is why the data literacy is important. Why data literacy is important? Okay, I will share my personal knowledge. Instead of, I also do some training. So besides my regular activities as a lecturer, I also invited to as a speaker in some training workshop and also consultant, something like that. And I have some experience that maybe one or two of them I would like to share with you. Yeah, so you have more perspective about how or why the data literacy is very important. Okay, the first case study that I want to share is it was happened back in 2019. When I was in a training about owner estimates. Owner estimate is actually there is kind of project owner, and they want to estimate the cost for the whole project before they offer another kind of another organization to have a bid, something like that. So back in 2019, there was a purchasing department of a government-owned company, or in Indonesia, you're familiar with BUMN. So the organization currently, at that time, they were currently developing an owner estimate for 2020 project plan. And then one of the team member of the purchasing department told me that they have difficulties on estimating 2020 province standard wages for East Kalimantan, or in bahasa you familiar with UMK, upah minimum UMP, sorry, upah minimum provinsi, yeah, or I just translate it freely to province standard wages for East Kalimantan. So that case, that problem has become a very big obstacle for them. Has become a very big obstacle for them to develop the owner estimates of the 2020 project plan. But actually, if they check further, yeah, if they see the data and they collect the data and process it, what they can find, and I showed them, yeah, on the spot at that time in the training, I showed them, okay, I had the data, I found the data from the internet, and I plot the data just using Microsoft Excel, and I use a simple forecasting method. It's a very simple forecasting approach. And the result of the forecasting showing that the upper confidence bound is about 2.93 million for the standard wages in East Kalimantan. Yeah, but when I verify a few weeks later, the government of East Kalimantan published announcement about the province standard wages, and their number is actually not far from the result of the very simple forecasting. It was 2.98 million. So actually, once again, this is one case that showing why data literacy is very important. And the second one, but I will not give you the solution. The solution actually, at this time I still keep it, and I have the data. If you're interested, you can contact me and we can share or discuss about how we solve this problem. So there are two mining companies. Yeah, one is kind of oil mining companies, and another one is, as I remember, is gold mine companies. And they're facing a similar issue. So what they need to do is they need to reduce their inventory value. Yeah. So the KPI for their inventory value is maximum is approximately $25 million US dollars. That's the KPI. And for some reason, their inventory value is above 25 million. Can you imagine $25 million is about 350 billion rupiah. Yeah. And there's like kind of capital that they have to, the company should provide only for a spare part inventory, only once again for a spare part inventory. But the question is, can they use their data to do so? Yeah. So if you're interested with this case study, you can contact me. We can do like a joint research. I have the data already with me. Yeah. But the question is, can they use the data to solve that problem? I already have some framework for this case study, and we just need to execute it. All right. Let's go back to the literacy for the better world. So as you remember that the second literacy for better world is system thinking. Yeah. So system thinking is once again is a mindset to think, communicate, and learn about system to make the full patterns clear. The key word is there, pattern. How we can see the patterns more clear or clearer. The question is how we find pattern in big data. Yeah, how we can find pattern in big data. Sorry. Okay, there are two main keywords actually. So if we talk about big data and pattern actually, what we need to see first, what I need to see first is what is the business interest, because approach I'm from industrial engineering, so the big data usually industrial engineering is not far from how we solve problem. This problem is actually kind of business interest. So if we want to see the pattern, we have to see what is the business interest. Okay, I will discuss that further on the next slides. So for you who has deep knowledge about big data, it's okay. Maybe it's a refreshment for you. But for you who has not yet familiar with big data, yeah, this is a small introduction about what's big data. So big data is actually the data who has what people usually call 6 V's, the very big 6 V's. Big in volume, variety, velocity, veracity, value, and variability. Yeah. So the resource of big data could be from many sources, but because of the big of the data, yeah, if you see that the growth of data, the most data collected from any available resource is actually more than or about 80% is actually unstructured data. And it's growing very fast. Yeah. From, you can see from 2013 to 2017, the growth of unstructured data is very fast compared with the structured data. Yeah. If you see there on back in 2013, yeah, maybe the unstructured data is about twice compared to structured data. But in 2017, yeah, the unstructured data, the volume of unstructured data could be 4 or 5 times more than the structured data. So this is also if I can present you the resource or the source of the growth of data. So the source of the data, it could be from enterprise data, from voice over internet protocol, from social media, from sensor and devices. Yeah. So if we go back again to the issue about the industrial revolution 1.0, where machine talk to machine talks to each other, it means that between them there are sensors, between them there are devices that enable them to talk to each other. Yeah, there's also the source of the data. Okay, so what I want to highlight here is the second one, the social media. Yeah, I would like to compare what happened in the internet in minutes, 2019 and 2019 and 2020. Yeah, because 2020 is actually we are now having a pandemic. So in some account of social media, in some usage of the social media, it was growing very, very significantly. As you say, if you check there, for example, in Instagram, yeah, Instagram back in 2019, there were about approximately 350 scrolling Instagram, but back in 2020, it is like double, as well as Twitter. Yeah, so social media is one example of the growth of the unstructured data. So this is also the characteristic of big data. Yeah, can you compare the characteristics of big data? If you say there from the archive, kemudian documents, business apps, media, social media, and something like that, compared with the in terms of the velocity, variety, and volume. Yeah, in terms of the velocity, variety, and volume. Okay, this is how social media is used in Indonesia. Yeah, some of you may be already familiar with this figure. Yeah, in Indonesia is currently there are 160 million users, active social media users in Indonesia. Okay, and this is the data about the most active social media platform. Yeah. Still in Indonesia, YouTube is #1, as well as in other countries as well. Yeah, and then what I use here is actually Twitter. Twitter is number 1, 2, 3, 4, 5, 6, number 6, or approximately 52% of the whole the user of social media. And It go back in January, or it was in January 2019 and go back in January 2020. The use of the Twitter is what is it? Increased from 52 to 56%. This is the data of the user of the Twitter itself. Okay. So actually, what's the big data analytics? Yeah, because I mentioned that the social media is one example of unstructured data. But actually, the unstructured data is very useful to enable us to solve the business interest of the problem that we face at this time. All right. So actually, how we process the data. The big data is kind of, especially the unstructured big data, is kind of random data. And the big data analytics is how we make a pattern. How we make a pattern, and those kind of pattern can be useful to make a decision. Yeah. So like some people already discussed this issue, that in the big data analytics, yeah, some skill that needed could be mathematical, IT, could be statistic, or combination of those three. But actually, there are one very important skill that enable the result of big data analytic into decision. Yeah, this is that maybe some people has not yet aware. Yeah, the person who we call as domain expert, who understand the data and who understand the business environment. In some issue, we call this domain expert as a storyteller. The storytellers is sometimes they don't have to need they don't need to have a very good skill at mathematical. They don't have to have like a good skill at statistics. But what they do need is actually they can read the result from data analytics and tell the decision maker what happened, or this storyteller. So the storyteller, their main job is to tell story about what happened. Yeah, this is a very important position, especially in the process of bringing the data into action. Okay, I will skip this one. Yeah, this is the definition of big data. Okay, and then I also skip this one. This is the stages, the step of how we do analytics in big data. We just did that. I will go back to the main issue, but this is also important. So this is the main benefit of the big data. Yeah. So if the company uses big data or use data to make decision, so actually 69% saying that using data or big data for making decision, it enables them to have a better strategic decision. Yeah, this is the result of kind of survey. And then 54% saying that the usage of big data can improve control of operation processing, and 52 saying that better understanding of the customer, and also it can help organization to deduct the cost. Okay, this is I will give you an example of how we can use the big data for a product development process. Okay, so actually product development process traditionally, yeah, is a very complicated process. There are several steps that should be done, from idea generation, idea screening, concept development, and so on, until we introduce the product and sell the product to the community. Okay, so yeah, once again, this is the traditional market research model. Yeah, So the traditional market research model was created to serve like a top-down product development process. So a product was defined through an initial market assessment. The spec were frozen, and everyone went to work executing the grand design, a typical waterfall process. So what is it? The process of traditional market research model, yeah, as you know that, usually they do a survey, and they do a survey, yeah. Not so long time ago. So what people did is they have a piece of paper, some sheet of paper maybe, and they go around to the market to find a very prospective customer and ask the customer to fill the survey. Yeah, and then by the development of technology, they start to use communication technology like telephone. They call their customer, and then with the development of IT, they try to do it online, but still using like a set of question or questionnaire. Okay, but what happened actually now? So in these days, what people want is highly individualized product. Yeah, the product that are continuously being updated and improved. Otherwise, the customer will say this product is outdated. Okay. All right. So how we can use big data in this case? All right. I give you an example of how social media can be used for the product development. Okay. I believe that you're already familiar with this product. Yeah. Maybe some of you also use the product. So this is the product that they aim to innovate the major and deodorant category. Okay, so like I mentioned, that traditionally, what the organization did is they carry out market research, ask questions to a selected audience or what they see as prospective customer, and try to draw a conclusion based on the result or the answer of the question. But this organization wanted to know what the customer are really saying about the product. Because if we ask them a question, maybe what they answer is just what they have to answer, not what is really happened. I believe that if you answer like a questionnaire, sometimes your answer is kind of like normative answer. So what should be the answer, not what really happened. But this organization, they want to see what the really requirement of their customer actually. So what happened is they look into the vast amount of data, not just select the number of participants in a focus group discussion or from questionnaire. They look their communication either in the blog, in the website, in social media, and something like that. And if you see that, what they found is actually they discover that there's some issue with the product. There's a residue that sometimes is difficult to remove. Yeah, if you maybe back to a few months or a few years ago, if you use some particular product, this type of product, it left some residue on someone smiling there. It means yes. So there are some residue on your clothes, and sometimes it's very difficult to wash. Yeah. And it needs a very special treatment to get rid of the residue. There's a problem that what is it that said by customer. And then this organization saw a very big market opportunity, and the result, they become a pioneer on this kind of development of the product. Okay, so I give another example of how me and my friends did. Yeah, if you see there on the top right, yeah, that's the tools that we used. Yeah, so this is the big data analytics for coronavirus event in Indonesia. It's go back to February or March this year. Yeah, early this year. So apologize for you who has from overseas. Yeah, I forgot to translate it to Indonesia because it's a bit difficult because what we use, the keywords is actually in Bahasa. Yeah, but I will translate it for you. So this is the graph of how we read the public emotion related with coronavirus in Indonesia. So the keyword that we use, if you check there are some words like pemerintah, pemerintah is government. Indonesia is very clear. Pemprov is a province government, gubernur is governor, wali kota is like a mayor, kabupaten is like a city or something, yeah, and province is province. That's the keyword, yeah. And then the filter that we use, yeah, for the data collection is corona virus, COVID-19, COVID-19, coronavirus, and virus corona. So virus corona is coronavirus in Bahasa. And mostly the social media platform that we observe is mostly from Twitter. And you can see there the public emotion related with this issue. So it was done when the government announced at the first time that coronavirus was in Indonesia. It's about back in March, as I remember, yeah, in the mid of March. So after the government published that information, yeah, we need to know how is the public emotion related with that announcement. And actually, the result, you can see that most of the emotion of the public is related with surprise and trust issue. Trust issue could be trust issue to the government. Yeah, but some of them also feel anger, yeah, fear, anticipation, and so on. And so, how we do that? We do that by reading their social media. Yeah, this is the example. Once again, apologize for those who not in Indonesia, yeah, because this research was done in Indonesia. So I'm afraid that if I translate it into Indonesia, it will change the essential of the research itself. Yeah. So this is the emotion analysis related with trust, and then this is related with the surprise. So actually, some of them feel surprised because they don't they think that because if we go back in January and February, some people is really believe that coronavirus will not become a pandemic in Indonesia because the Indonesian people naturally they have like a immune against the coronavirus, like a natural immune against the coronavirus. But actually, that kind of belief was wrong. Yeah, was wrong. Okay, this is also how we read the people related with anticipation, and on your right hand side is the example of their comment, and also this is the example of they feel fear about this event, this coronavirus pandemic, and also this is the anger. Okay, so another issue that I want to share is about omnibus law. Omnibus law. For you are in Indonesia, omnibus law was a very hot issue until a few weeks ago. Yeah, and actually, what we want to know is how is the actual what people actually think about omnibus law. Yeah, and this is the SNA, the map that we can see from the omnibus law. Yeah, so you see there's kind of maybe you see kind of rainbow or galaxy or something like that. So actually, one dot, yeah, one dot is one account, one account. So if you see there are a lot of connected dots, it means that in Twitter, one person retweet another account, one account retweet another account. So, and they develop kind of network. And you see there, there's the map for those people who support and disagree with the omnibus law in Indonesia. Yeah, I myself, yeah, because I don't have any idea about omnibus law actually. Yeah, so I need to read it more further because yeah, we are as a scientist or as an engineer, we have to see the big view. Yeah, I will not provide my personal opinion about this. I just present the data. Yeah, I'm not in the side who support. I'm not in the side who disagree with omnibus law, but I just present the data. All right, and this is the hashtag. Yeah, the hashtag. So actually, there are some hashtag that also agree with omnibus law, but most of the hashtag used is against the omnibus law. Okay, the research that we have done is actually use what we call as drone umbrella academics. Yeah, drone umbrella academics is like social media data analysis. Yeah. So for all of you, for all of you who are interested in drone umbrella academic, yeah, you can join us. Once again, you can join us. You can see the website there, dea.uii.ac.id. Yeah, it's free of charge. It's really free. Yeah, it was provided by Universitas Islam Indonesia. Yeah, but for this academic version, apologies here is running very heavy. Yeah, so maybe give some noise. So for you who are interested in reading the data, the unstructured data, in the unstructured social media data, especially in Twitter. Okay, you can join us. This is free, yeah. And then this is I will show you the dashboard, and this is how it works. Yeah. So anyone can join. Either you are a user, researchers, lecturers, journalists, blogger, podcaster. Anyone can join. Yeah, for free. But what we need from you is actually you just publish the result of your analysis for the public users from any publication, paper publication, or just in your website, the social media, or just like infographic, something like that. Just share your result. That's it. That's only the requirement to join us. And at this time, yeah, back in 6 months ago, I don't have the update until today, but we have about 285 projects using Dogma. And this is also the example. And also, this is our member. Actually, we still have about 600 and continue. Could be now moved to 700 as I remember. And this is also the publication using the Dogma Academics in Google Scholar. Okay. And I was requested by Bu Anissa to have like kind of closing remark. So actually, go back to the figure about the importance of the big data literacy. So actually, the resource to solve the problem is actually lies around the problem itself. Once again, the resource to solve the problem lies around the problem itself. So what we need to do is we need to find the pieces of puzzle and solve it. Yeah, I just make this this quote, yeah, few minutes ago before I start the presentation. So yeah, so don't get what is it don't get down with the problem you face. Yeah, the resource of to solve the problem actually lies upon the problem itself. Okay, I think that's all for today that I can share with you, and I return the opportunity back to Bu Anissa. Thank you.
Okay, thank you very much, Mr. Winda, for your interesting presentation. I hope all of the participants can understand what Winda said and get new knowledge, yeah, from the talk. Mr. Rinda has already explained to us related with the big data analysis for social media data. And as we know that today data grows so fast, and usually we use term of big data to explain the data that has characteristic of with the volume, velocity, variety, veracity, value, variability, and so on. Actually, there are a lot of V to characterize the big data. And the big data itself can help us, if we do the analysis in the big data, help us to generate a sound decision, like what Pak Winda has already shared to us. It can help us in product development process. It can also use in sentiment analysis, like in these current issues like coronavirus or omnibus law and so on. Okay, thank you very much, Pak Winda. And now time for question and answer. We have already several questions from the participants here. For the first question from Klister, yeah, from the University of Technology Yogyakarta. According to you, to what extent have machine learning been used in Indonesia, and whether the use of machine learning in processing data is better than deep learning, especially in overcoming the impact of COVID-19?
Okay, that's tricky. Because as I remember, this question was asked to the previous presenter, but actually I try to answer this one. So to what extent the machine learning being used in Indonesia, and whether the use of the machine learning in the process saying that is better than deep learning. To be honest, I don't have better understanding, and I don't have a very deep understanding about comparing the machine learning and deep learning. Yeah, so I'm not sure, but maybe if you like, I can ask my colleagues about how the difference between machine learning and deep learning, especially in overcoming of the impact of COVID-19. As I know, yeah, after the discussion with my colleagues, it's not, unfortunately, machine learning and deep learning has not been used for overcoming the impact of COVID-19. What they have been used is to model the pattern of the what is it, the COVID-19 pandemic. Yeah, as you remember, maybe one university in Singapore did that, but it's not it's not valid either. Yeah. They predict that it will be finished maybe by the middle of this year, but actually it goes on until that. Yeah. So apologize for this liar. Yeah, I cannot answer your question better than that.
Okay, thank you very much. We will also send this question to Professor Anirban, and hopefully they can answer it in email. Okay, thank you very much, Pak Winda. For the next question, how we can make a pattern and organize this unstructured data?
Okay, thank you. That's a good question. How we can make a pattern and organize on the unstructured data. So what we have done, especially in social media, yeah, it's just similar like we actually what is it? We try to determine a correct keywords actually, because unstructured data, if you see that, it's very unstructured. So how to make the pattern? First, what we need to do? we need to determine what is the business interest first. If we understand what's the business interest, from the business interest, and then we need to find a correct keyword that enable us to see what's the basic purpose of the people, what they really want to say in their social media account. Because if we use, for example, if we want to see either people afraid or not, if we only use afraid, maybe we will come up with everybody's afraid. Because in Indonesia, the opposite of afraid is not afraid. Takut and tidak takut. So if we only use one keyword, which is afraid or takut, it will make us going nowhere. So actually, to make the pattern and organize the unstructured data, the choice of the keyword on the unstructured data is very important. This is my experience in social media. Okay, do you understand?
Oh, I'm sorry, still mute. Thank you very much, Pak Winda. For the next question is from Fadlan Wisahasibuan. Managing the data in social media is quite tough to handle by person with number, beside the cost, sales, etc. The case in social media is how to keep the trend in our social media is staying high, how to generate the interest of our viewer or customers to be good product and content. Oh, sorry, it's black out here. Okay.
I think Fadlan can ask me personally.
Okay.
He was my student in IP, as I remember. Fadlan, how are you? I hope she's still there. And why you give me a very difficult question here. So the managing the data in social media is quite tough. Yeah. So what was the main question? Do you understand? How to generate the interest of our viewer to be a good product or content? I think it is the issue of marketing, not big data and social media. You understand? Because the question how to generate the interest, right? But it is possible actually. It is possible. So what we can do is actually we can see, for example, this account who has a good interest of viewer, yeah, and that account we can develop kind of unstructured data on that account, based on our business interest, yeah, and see the pattern why those people or viewers interest to that account. After we found something, the result of our observation can be applied to other accounts in order to generate interest for a particular product or content. Thank you, guys.
Okay, thank you very much, Pak Winda. Maybe Mr. Fadlan can ask Pak Winda directly if you still have a question related with this topic. Yeah. Okay, for the next question from Muhammad Naufal Alvareza, Prof. Winda, is there any way to validate the data taken from social media? For example, like Twitter, how do we know, or how do we filter out that the data we take is not from the buzzer? Wow.
Okay.
Thank you.
So it's difficult to see whether it's buzzer or no. Yeah, buzzer or no. But there are some websites that you can use to identify whether it's a bot or no, it's a robot account or no. Yeah, there are some websites that you can use to identify a particular account is a robot or is a real people. All right. So how you identify buzzer actually? How identify buzzer? If you see on my slide, you see this maybe like in SMA slide, yeah, slide that look like a galaxy and rainbow. Buzzer usually they talk to each other. They just make their own group. Yeah. In our tools, buzzer like they just talk to each other. they will not retweet other people's account. They just retweet their own. Yeah. So what we observe actually issue about palm oil, kelapa sawit, issue about what is that, omnibus law, and any other issue. We can identify usually this is agree, this is disagree. But there is another pole that we identify as a buzzer. Why? Because they just talk to themselves. They tweet one to another. So that's how we have experience to identify buzzer. But the most interesting thing is how we validate the data from social media. It's difficult because people usually they want to be perfect in social media. And that's the big issue. So sometimes social media and the real world is pretty much different. But if we want to identify the bot account or the buzzer account, it could be done. But to validate is as difficult as when you give a questionnaire to your customer and want to see either the answer is the real answer or just want to make you happy. Yes.
Okay, thank you, Pak Winda, for your answer. And then for the next question from Mr. Biswajit Biswas. Sir, what platform need to use to analyze the emotions as like Java or Python or R?
Emotion.
And also,
yes, you can join the Academy, but unfortunately, because it's academic version, we just able to read the emotion from Twitter. But I think the user of Twitter is a lot. So I think it's good also for you to read emotion from there.
okay. And this is still from Mr. Biswajit Biswas.
Okay,
I don't know, is this a question or is this a statement? So we can analyze emotion feedback using this technique. Yeah, of course.
Yes, we can.
Yes.
Yes, we can.
Okay, okay. I think that's all of the questions. Thank you very much, Pak Winda, for answering those questions. And also thank you very much for your great presentations. And it was a pleasure to have you as our speaker today. So I think this is concludes our second session of our webinar today. Thank you for all of your attention. And we hope all of us learned something and enjoyed this presentation. Okay, ladies and gentlemen, I think this is the end of the presentation session. Alhamdulillah Rabbil Alamin. Thank you very much for all of your attention. And me as the moderator send my apology if there are some mistakes conducted, and hopefully we can all learn together, and hopefully we can still help safety and in best conditions. I mean, and I will give hand back to Mbak Safina for continue this session. That's all for me. Assalamualaikum warahmatullahi wabarakatuh. Thank you very much.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mr. Winda Nur Cahyo, and also our moderator, Mrs. Anissa Uswatun Hasanah, for today's presentation, and also for Professor Anirban Mukabadi has already left because he has several things to do. He would like to say sorry for all of us because he cannot join until the end of this Studium General. And next agenda is the closing speech from Mrs. Ira Promasanti Rahmadewi as the Secretary of Industrial Engineering International Program Department. For Mrs. Ira Promasanti Rahmadewi, time is yours. Maybe the committee will help Mrs. Ira to share screen or something. Time is yours.
Ya, Alhamdulillahirabbil alamin. Finally, this webinar series in this term are about to an end. IP still has one more topic to discuss. However, it is temporarily limited to international program of industrial engineering Universitas Islam Indonesia students. International program industrial engineering students right now are engaging a project in collaborations with Deakin University Australia and PTI company in Vietnam. The students are building face attendance system, implementing artificial intelligence to build a face attendance system using a small camera and also Internet of Things edge device. Because due to COVID-19 outbreak, the system should be able to secure the social distance and also the people wearing mask before entering a place. Yeah. The models of face attendance systems are face detection model, face recognition model, and also mass detection model. The objective of this project is actually to explain and practice the process of building an artificial intelligence product, and also professionally to perform artificial models using Python and also TensorFlow, and also get familiar and make use of tools in software project development such as Git, Jira, yeah, server share. Okay, this is I just want to say that industrial engineering students are able to build a project with artificial intelligence system. And also remember, international program industrial engineering still has plenty of challenging topics or issues related to Industrial Revolution 4.0 and also business environment in artificial intelligence era in the upcoming term with amazing speakers. I wish you always stay tuned and join our webinar talks because IP will definitely invite you. Okay, if you want to get in touch with IP, just visiting our website, Instagram, and also line, email, hotline service using WhatsApp. And also IP has YouTube channel. Yeah, IP has YouTube channel is also serving interesting contents for you to study. Okay, thank you very much for your joining. By saying Alhamdulillah, I officially close this autumn 2020-2021 webinar series. Hopefully, you are always in a healthy and good condition. See you again in the next international program, Industrial Engineering, Universitas Islam Indonesia Talk. Wassalamualaikum warahmatullahi wabarakatuh. Bye.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mrs. Ira, for giving a closing speech for our Studium General today. And I would like to say thank you very much for your enthusiasm and also your time and effort and everything to hold this Studium General. Thank you very much for amazing speakers. I hope we could get new knowledge to boost and increase our insights about big data, which is related to Industrial Revolution 4.0. And we're living in this kind of city right now. Me, as the name of the committee itself, would like to apologize if there is some mistakes in our attitude or words. And also, as the Master of Ceremony in the Studium General, I would like to say goodbye and see you in our next event. And make sure that you guys put your notification on our Instagram, so if we have updated, you will notify soon. That's all for me as your Master of Ceremony. Thank you very much for your attention, and wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much, everyone.
Thank you. Sudah, everyone. Pamit dulu ya.